Thanks once again. I'd like to make this talk much more focused on Alzheimer's disease and with a little bit about frontotemporal dementia. And really, why should one have an interest in tau in Alzheimer's disease, since everybody seems to think that amyloid is the main driver. So as the disease progresses, we now recognize these stages. There's a fairly long preclinical prodrome where we need tests like Helen's to pick up the earliest cognitive changes. Then there's prodromal, which is now the term for MCI, mild cognitive impairment. And there is measurable cognitive impairment. In mild AD, you have impairment on cognition, functional impairment, so activities of daily living and so on, and global clinical evidence, moderate and so on. So in severe AD, really the behavior becomes abnormal. This is kind of stereotyped. People differ individually in how they behave and how the disease interacts with their personality, but this is the broad profile. This is a commonly quoted slide that comes from Jack et al. From Lancet Neurology, and it envisages that the amyloid changes seen here in purple are really the very first thing that goes on or is abnormal, followed by PET scan, followed by changes in tau, and then followed by MRI changes, and then the cognitive impairment. And there are many talks, particularly by people who favor the amyloid theory of AD, that will often start with this presentation. However, if you look quantitatively, the data is not quite like that. Here is a study from Donahue et al. From last year, from the ADNI program in 700-odd cases. And this is a plot of how abnormal people are on their CSF markers. So you can see that they, at 15 years before the onset of dementia, they progressive, somewhere between minus 15 and minus 10 years, they become more abnormal, and then even more abnormal, really quite late in the disease. The point I want to make is that likewise the tau abnormality sets in at about the same time. That's the key point I want to make. If you look at the rate of change, the slope divided by the standard deviation, that makes it even clearer that the two events are very closely associated. This is another study from Buckhave et al. In 2012. You've got to read this from right to left. So here are controls. MCI, this is looking at those who are converting from MCI to AD 5 to 10 years before their conversion, or 2.5 to 5 years before conversion, or less than 2.5 years. You can see that there's a progressive increase in the tau, and the amyloid goes down at about the same time. And the only point I'm trying to make is that they are coincident and one does not precede the other. This is a very interesting study that came out last year as well, and the year before, looking at a plasma marker for tau. This uses quite sophisticated technology to detect very low levels in the picogram per mil level. But in plasma, and this would be wonderful if you could go to your GP and just have a blood test to see if this pathology is going on. So you can see people with MCI are already discriminated at an early stage, and then in early or mild AD, there's even better discrimination. And these plasma markers are highly correlated with hippocampal volume and with logical memory. So hippocampal volume as measured by MRI and logical memory. So these sorts of tests hold high promise as ability to intervene early in the disease process. So I showed this slide before, and I just won't spend time really, but just to say that the tau aggregation begins as oligomers and forms fibrils and so on. And that essentially destroys the pyramidal cell communication system in the neocortex. So all cortico-cortical associations break down. So another interesting technology that we're looking at that I won't be speaking about is the use of EEG to look at that breakdown of cortico-cortical connectivity, and there's some very promising data on that as well. So I also showed this slide before, that the aggregation is prion-like, that is, that tau is progressively converted into a proteolytically stable form, which acts as a nucleus for more tau capture and more creation of more proteolytically stable aggregate, and the process is endless. And I also showed this before, that contrary to firmly entrenched opinion in the field, that amyloid is somehow on a straight line of causality between amyloid pathology and tau pathology, the situation is probably not linear at all, that the main driver is probably an age-related impairment in autosomal lysosomal processing. So it's a deficit in autophagy. It's an inability of cells to clear rubbish, essentially. Now, the biggest consumer of the rubbish clearance service in a nerve cell is actually mitochondria. Mitochondria turn over very rapidly. One mitochondrion lasts maybe 100 days and is an obligate turnover substrate. And what happens in the aging brain is that system becomes impaired. And the marker of that impairment is the accumulation of lipofuscin deposits. So lipofuscin, which is generally regarded as an aging pigment seen in neurons and in myocardium. So in other words, non-dividing cells, so pyramidal cells last for 70, 80 years, a single cell. You stay with the same cell population forever. Inside that, there is rubbish, accumulating rubbish that is autofluorescent. It's called lipofuscin. But when you analyze it chemically, it's essentially 3 mitochondrial membrane proteins, porin, core protein 2, and complex 3, and also a fourth one, ATP synthase subunit 9. So these proteins, it turns out, co-purify with very pure preparations of tau. So it's likely that this is the real driver, the thing that converts tau from a completely normal molecule into this aggressive pathology disseminating species, is a primary abnormal binding event. So most likely all that happens is that tau gets stuck on something, gets stuck on rubbish, that causes a primary change in conformation, which exposes this high affinity repeat domain, this high affinity binding site. Now it turns out that binding site has greater affinity than the tau tubulin binding site. So once it gets going, all of the tau gets diverted down this pathway. And I've shown this before. What I want to concentrate now is more on the spread of the pathology. So I said before that it spreads out of medial temporal lobe structures into isocortex via a process that is not well understood at the moment, and that this spread that is measured by Braak staging is highly correlated both with cognitive impairment and with functional imaging impairments. What have become available in the last year and a half, 2 years, are specific PET imaging markers of tau aggregation. So the one on which most work has been done is from Tohoku University in Japan, and you can pick up tau pathology in the medial temporal lobe. And there was a report last year at the AIC meeting which showed that in clinically normal elderly, there is an association between neocortical tau aggregation, as measured by PET, and decline in memory performance, and likewise the same marker. In another study, there's this very lovely correlation between the signal, i.e. the tau aggregation signal, and decline on Mini-Mental score. So as your signal goes up, your level of impairment also increases. This aggregation process, I said, is autocatalytic, and we've done a lot of studies, and this is just a summary diagram which will be coming out in a book later this year on development of treatments approaches for Alzheimer's disease. But what this shows is that the quantity of aggregated tau measured in picomoles per gram is really, really an exponential process. So it's a one-way train trip, one track. And what we see as the transition from Mini-Mental 30 to the beginnings of impairment is simply this transition into this rapid exponential phase, but there is a long prodrome of progressive aggregation. You can actually map this process quite well, bringing together a whole lot of data that we obtained from the prospective cohort that we analyzed in Cambridge. And this is kind of a unified view of everything from a tau perspective. So here is the quantity of neocortical tau in parietal and temporal lobe measured in nanograms per gram. Firstly, this is a logarithmic scale. So in other words, we're trying to capture this huge range that's going on here. So it looks benign, but this is actually a logarithmic scale. Along here, you have time since Braak stage 1. And I'll talk more about the timing of Braak stage, but you can convert this from various big studies that have been done, how long it takes to progress. So this is the corresponding Mini-Mental score. So you can see that there is a long period of decline from 30 to 25, and then progressively each 5-unit decline in Mini-Mental occurs more rapidly and is associated with bigger and bigger changes in levels of aggregated tau. So the next question is, what is the threshold at which tangles appear? So what we're measuring here is actual quantity of aggregated tau, and this defines the first threshold in which the aggregated tau actually becomes visible as a tangle. And then there is a second threshold where it becomes of a sufficient density to kill the neuron and simply leave behind what's called a ghost tangle. And this happens at different rates in different brain regions. So in a person who has a Mini-Mental of 25, they are already at the tangle stage in their entorhinal cortex. They are just moving into tangles in their hippocampus, and they don't have any tangles at all in the neocortex. That's not to say that they don't have aggregated tau. It's simply that different brain regions at different points are at different points in the evolution of the pathology. I showed you before that based on cerebrospinal fluid data, it's not at all clear that amyloid precedes tau. And the neuropathological data is really quite decisive. This is a large study from Braak, and he's measuring the development of different stages of amyloid pathology. He has a staging system for amyloid, and different stages of tau pathology as measured by a tau marker. And you can see the tau is definitely occurring before. This has been confirmed in another very large study from the Mayo Clinic in the US. This was in 2,300-odd subjects. The Mayo study is in about 3,500 subjects. Exactly the same thing, that amyloid is not predicting cognitive decline, that people who have no amyloid in their brain already 50% or so are already at Braak stage 2 in a very big prospective study. So it's decisive. This was already shown in 2011 that the tau pathology is appearing before the amyloid pathology. However you look at it, it really is not the case that amyloid is coming first, at least in terms of pathology. In cerebrospinal fluid, they appear to be neck and neck. I said, how does one convert tau into, or Braak stage into time? And the key to this was a very large study published by the Braak group, Omics et al. 95, where they looked at 847 post-mortems aged 45 to 95 with 17 cases per year of life. A really beautiful study, unselected cases. Here, what we've done is taken their data and plotted essentially a Kaplan-Meier survival plot, which is the probability of transition from nothing to Braak 1 or beyond, transition to Braak 2 or beyond, Braak 3 or beyond, Braak 4 or beyond. This says several important things. Firstly, everyone gets it in the end, essentially. Whether it's visible clinically or not, everyone gets it if you live long enough. What's more important is to get a sense of how many people are at different stages. So we applied this data to the Canadian population data for 2010. I did it for the Canadian population because we're in Toronto, or actually Toronto, as it's pronounced here. But the European data will simply be 10 times this, roughly, and actually more than 10 times, and the US data will be about 7 times these numbers. But I'm just pegging it to Canada because we're in Canada. So in Canada, if we look at the over 45 population, firstly, about 50% will have some degree of tau pathology in their brains. So this is the age profile of those who are at Braak 1, and you can see the peak is at about mid-50s, but it can start quite early. That about 11% are at Braak stage 2, 10% are at Braak stage 3, and 5% are at Braak stage 4 or beyond. Now, this is about the numbers that you have for AD. This is the typical number, 5% of the population will have, of the elderly population, here I'm sort of widening the scope a little bit, will have visible dementia. However, what this is striking about this is that the population at Braak 2 essentially becomes the population at Braak 3, but 10 years later. So in other words, you can have Braak 1 and it won't hurt you, so to speak. But once you go to Braak 2, you're done. And those the only reason this number is less than this number is because they start to die. People start to die. So coming back to this graph here, what does Braak 2 mean in practice? Braak 2 means a mini-mental of 30. So in other words, we really want Helen's kinds of instruments or whatever other tools that we can have to detect the people who are on this trajectory, this lethal or pathological trajectory. So in other words, when someone goes to the doctor and says, I'm not quite right, I don't seem to be able to do what I used to do, and if the doctor goes to the trouble of doing a mini-mental, or let's say even a more sophisticated test, very often they'll say to the person, there's nothing really wrong with you, and they're dead wrong, because quite likely that person is Braak 2. That's why we really need much better markers of early stage disease. So when does Braak 1 begin? And this just summarizes the ages. So it can begin at 38, but peaks at 58. Stage 2 can begin at 48, but peaks at 63. Stage 3 can begin at 53, but peaks somewhere between 68 and 78. And stages 4 to 6, which is real dementia, can begin at 43, but peaks at 88. If we then do the fast forward on this analysis and use the UN population figures for 2010, 2030, and 2050, the numbers themselves don't matter, but just the shape of the graphs tell you the demographic transformation that is occurring in all societies, and it's even more dramatic in China and Taiwan and in Southeast Asia, where there is a rapid economic development going on, and a rapid aging of the population. But look at the BRAC 4 to 6 today, more or less today, 2030 and 2050. This is a huge, huge care burden that really has to be arrested somehow. Okay, so this just summarizes what I've been saying, the scary statistics, that the over 45 population, 51% will have some degree of tau pathology. Half of that 50% will be at BRAC 1, but 10% will be at BRAC 2, 10% at BRAC 3, and 5% at BRAC 4 or beyond. They're very simple figures, but they are really terrifying in terms of the demographics of the disease. So my big take-home message is tau is bad. It's an endogenously generated infectious prion-like process that won't go away by itself. And irrespective of the 20 years of amyloid versus tau debate, this is a completely valid pharmaceutical target. Even if amyloid is right, even if amyloid is wrong, actually I don't care, but I'm quite sure that a tau-based treatment has a place in the world. Okay, so that takes us immediately to this. So I showed you before this molecule that is actually an old molecule. It's been in pharmaceutical use for about 100 years. It's conventionally known as methylene blue. It's methylthionynium chloride. And this is its molecular structure, and this is our novel stabilized reduced form. So what does this molecule do? And I showed you in my previous talk that essentially it dissolves the oligomers and dissolves the aggregates. So it neutralizes this propagation of the disease, and it takes away the element which is the autocatalytic turbocharger of tau aggregation. So it converts proteolytically stable tau aggregates into proteolytically susceptible monomers by melting, that's the term that crystallographers use, the paracrystalline tau polymer. This is the data which we recently published from our phase 2 trial. It was a fairly large study in 321 subjects, and the primary efficacy endpoint was ADAS-Cog change at 24 weeks, according to severity. So in our primary analysis, we split, and we pre-specified the split into mild and moderate. So you can see there's no placebo decline in the milds, but in the moderates there was decline, and at the optimal dose, which turned out to be 138 milligrams per day, we arrested progression. Likewise on the global measure, CIGIC, and on mini-mental. So these effects were all big and statistically significant. A mini-mental benefit of 3.79 is very big. The Aricept produces a benefit of 1 mini-mental unit, roughly. So this is 3 to 4 times better than Aricept. These are the p-values. These are the corrected p-values because you've got to correct for multiple comparisons. The big upset in this study was that the high dose failed. So it was really the middle dose that turned out to be the best dose. And it took us several years to get to the bottom of why that was. And that's really why we've gone on from the old form of the molecule to the new form. The absorption of this charged molecule depends on its being first reduced in the gut, actually the stomach, into an uncharged form which can then be absorbed. And that doesn't really happen very well either in the presence of food, and we made it worse by the particular formulation that we used. So what I've plotted here is the available dose versus the nominal dose. So you can see that actually the middle dose was the highest available dose. If you then plot the available dose against the effect on brain scan, which I'll show you in a moment, or the ADAS-Cog effect size at 50 weeks, or the ADAS-Cog effect size at 24 weeks, that's the thing that is correlated with benefit. So it's the available dose. We had a wonderful brain scan result shown by HMPAO SPECT, which I showed you before, is correlated with Braak staging. This is the 8 brain regions, the decline that occurred over 6 months in the placebo arm, and here are the low dose, the middle dose, and the high dose, and I've just shown you that the high dose really failed to deliver adequately. And here, whereas we saw the clinical effects in moderate AD, we saw the scan benefits in mild AD, and I'll reconcile that for you in a moment. This is a statistical parametric map, which is asking the question, in what regions was there a prevention of decline? So we can see this here in using pre-specified regions, but you can simply use statistics to map out what are the regions where decline was prevented. And the answer is it's the Braak staging regions. So it's the medial temporal lobe structures and the temporoparietal regions. So in other words, the drug is preventing decline in the Braak regions. What was really surprising was that the scan effect at 24 weeks, where there was no clinical benefit, remember, predicted with a very high degree of accuracy the future clinical benefit at 50 weeks. So it's really, so in other words, the drug is producing a change which takes time to be manifest clinically, which is wonderful because it's exactly what you want with a disease-modifying drug. This is the data at 50 weeks showing only the most effective dose of 138 milligrams per day. And here is the control arm. Here are historical placebo declines, and here is the group at 138 milligrams per day, using an analysis that is approved by the FDA and which we will be using in phase 3. So we basically produced an arrest of progression by 85%. In other words, that people have 85% less decline with a standard error of 30%. So that was the result of phase 2. We also did a pilot study. I mentioned before that TDP-43 aggregates in frontotemporal dementia. So we did a very small pilot study in 7 subjects. This is the anticipated decline from subjects who have behavioral variant frontotemporal dementia and are at what's called KIPS stage 2, which means that they have definite MRI evidence of atrophy in frontal or temporal lobes. And here is what happened to people on active treatment. So you can see that we prevented decline, and even in 7 cases, the effect is statistically significant. In particular, the effect at 9 months is significantly different from 0. In other words, people actually improved. So we're now doing 3 trials in parallel. Yes, I'm finishing. So there's a study in 890 subjects, which is a 15-month study testing 2 doses, a mild study in 800 subjects, 18-month study testing the 200 milligram dose, and behavioral variant frontotemporal dementia in 220 subjects, and again testing the 200 milligram dose. So here we are just about to enter where are we? We're here, actually. So the last patient, last visit in this study will be at the end of this year, and we'll be announcing top line in the first half of next year sometime. So the conclusions: the presumption of temporal and etiological priority of altered processing of APP is unproven. It is simply a widely accepted conjecture. The genetic data for APP mutations being linked to AD can be understood as an accelerating factor in an underlying age-related process. There is no basis for the presumption that tau pathology is late stage, although it is often said. Intervention in the tau aggregation pathway is an entirely valid pharmaceutical target, irrespective of the beta amyloid theory. The phase 2 trials demonstrated the feasibility of treatment and efficacy, and the phase 3 trials are ongoing and will be available next year. And as I mentioned in my earlier talk, this has important implications for a range of other age-related neurodegenerative disorders. Thank you. So.
Questions?
Are there any preliminary data for BBF?
I'm going to have to repeat that.
Just the question is whether there are any preliminary data for BBFTD available from this multicenter?
No. I mean, it can't be. These are all double-blind studies. I can just tell you what my expectations are, but as you see, the behavioral variant dementia study will really only be in a position to report that mid next year. After, what has to happen is that last patient, last visit has to happen, then data cleanup has to happen, then you have to do the top-line analysis, and so on. So they're the timelines. I used to have a colleague at the Laboratory of Molecular Biology who said the difference between science and a detective novel is that when you turn over the next page, it's blank. Whereas in a detective novel, you can look at the last page and find out who done it. But science is like that.
It is very important to find an experimental model, animal experimental model for different stages of Alzheimer's disease and the dementia to find the possible treatment for each stage.
Yes. Yeah. I didn't have time to go into the transgenic animal data, but we published that. We published 2 transgenic animal models this year, and if you search under my name, or the first author is Mellis, Valeria Mellis, you'll find a report of that. But the proxy for early stage is simply younger animals. But these animals develop very clear tau pathology. And I don't believe that the treatments for different stages of the disease are going to be different, at least not in the tau landscape, because as I said, this slide here, this is a one-track train. This is a single-track train track. Yeah, it's all one path. So it doesn't matter whether you're pre-mini-mental 30 or you've just lost 5 points. They're just different stops on one train track.
We have a poster for modeling the different stages of Alzheimer's disease in response to time and the different doses of aluminum chloride. We make a model for different stages of Alzheimer's disease.
Okay, yes, well, I'll look at that. Yeah.
Thank you.
Okay, good.